thank you very much for a very nice introduction, and thank you so much for the MP and Max Birnstiel Foundation for this great honor. From my undergraduate days, I was very interested in the process of cell division and especially the process of mitosis. And the first description of mitosis was actually done more than 140 years ago by Walter Fleming. And to this date, it's probably actually one of the most accurate and the best description of it, where he observed these dark staining bodies, as he called, now we know them as chromosomes, being first aligned at the middle of the cells, and then being pooled and segregated to two daughter cells. What I find actually quite remarkable from reading this very, very old manuscript is that he observed this, what he called the thin needles, as you can see here. And he proposed that the thin needles actually attach to chromosomes and actively segregate them to two daughter cells. And it took another 102 years for Kirshner and Mitchison to show that such structures, now known as microtubules, actually exist, and that they can do the same work of actually pulling chromosomes to two daughter cells. Now we have much more sophisticated tools to study chromosome segregation, where we can label chromosomes in red directly and see how they're being pushed and pulled by the mitotic spindle, aligned at the midplane of the cells, all of the errors corrected, and then effectively rapidly segregated to two daughter cells. And the protein complex responsible for the attachment of chromosomes, shown in blue, to the mitotic spindle, shown in green, are known as kinetochores, and they're visible with small red dots at the tips of the microtubules. Kinetochores assemble at very specific locations on chromosomes known as centromeres, and centromeres are visible as the primary constriction points on chromosomes. In human cells, the centromere is epigenetically defined by having this unique histone variant known as the SNP nucleosomes. And SNP is really a subtype of a canonical H3 nucleosome. In addition to having a unique epigenetic mark, the centromere also has a unique DNA sequence, and it is made of many, many repeats of the 171 base pairs of alpha satellite DNA. The function of this kind of inner centromere is to effectively recruit the core complex of the kinetochore known as the inner kinetochore, which is a 16 subunit protein complex that directly recognizes the SNP, and during mitosis, it recruits outer kinetochore, another 10 subunit protein complex. Outer kinetochore, via its NDC80 subunit, directly attaches to the microtubules, thereby linking the underlying centromeric chromatin through the inner kinetochore, through the outer kinetochore, to the mitotic spindle. And so the first question during my PhD that we had was, how do kinetochores assemble specifically at the centromeres, and how can they withstand forces applied by the mitotic spindle during cell division? To do this, we've purified all of the components of this protein complex recombinantly, including the SNP nucleosome, reconstituted with native alpha satellite DNA, and we performed in vitro reconstitution experiments. And this is one example where we combined all of these components together with the SNP nucleosome, and then we see that all of the 20 subunits of this protein complex elute in a single peak with all of the components being identified. And I'm a structural biologist, so then we apply cryo-EM analysis to this process, which is a very intricate technique which can be summarized in this nice image. And this allowed us to determine the high-resolution structure of SNP nucleosome, which is effectively this big structure at the back, which bound the edge on to the inner kinetochore, also known as the secant, which sits in front. And this immediately revealed very striking features, such that the primary interaction site with the nucleosome was actually through this DNA shown in golden that goes through the main channel. Because we could achieve quite high resolution, we could actually assign all of the 20 subunits of this protein complex and see how they all fit together and how they're built. And we just start our journey at the back with DNA starting here, and DNA really wraps around the SNP nucleosome in a very canonical way. Then it threads through the central tunnel of the secant, or in the kinetochore, and particularly it's really gripped by these two subunits, SNP-LN, in the center. And then strikingly, what happens to DNA, it starts to wrap around this module called SNP-WSX, as if it was a nucleosome itself. It's effectively in the kinetochore, entraps DNA, and functions almost like a nucleosome in its own right. So again, the main emphasis is that DNA is really going through the secant, and secant topologically entraps this DNA. And then this linked DNA starts to wrap around this SNP-WSX module. And if we compare side by side the structure of SNP-WSX to the structure of a canonical H3H4 nucleosome, hope you can appreciate how similar they are, and that even the way that the wrap DNA is very, very similar. So secant really functions as a nucleosome-like particle. Then let me just come back to my initial question of how does the specificity of this inner kinetochore for the centromeric SNP nucleosome achieved? Well, we think there are two main factors. First of all, that secant really has to bind to the free linked DNA. So centromeric chromatin has to have a lot of free linked DNA going through it. And second of all, us and other groups, we see effectively only a single protein that directly interacts with the synphase nucleosome. If you just zoom in on the surface of the synphase nucleosome, we see this protein in yellow going along the surface, which is actually the same C protein, part of the inner kinetochore. And then it dips down and directly recognizes the protein in orange, which is actually here the synphase histone itself. So in this way, SNP can bind to the synphase histone in a very specific way. And then as a thread, it goes through the entire inner kinetochore, stitching it together and localizing it to the centromeric chromatin. So what about the second question? How can kinetochores withstand strong forces applied from multiple directions during chromosome segregation? And I think the answer to this question lies in the way that inner kinetochore interacts with the DNA. It effectively topologically entraps the DNA, serving as a topological device with a lot of protein mass all the way around the DNA, shown here in gray. And if you can imagine topological device, it doesn't matter which direction you apply force, be it from the top or from the bottom, your topological device will stay associated with the DNA. So far, I've been talking about the inner kinetochore bound to the centromere, which is effectively this left-hand side of the complex. But what about the outer kinetochore? So we applied similar principles where we reconstituted all of the 10 subunits of the outer kinetochore and determined its high-resolution structure. And in this reconstruction, we can see the high resolution 8 out of 10 subunits of the outer kinetochore. And the structure was also very intriguing. It provided some interesting insights into the assembly of the outer kinetochore as a whole complex. One of the striking features, at least to my mind, was that how rigid this entire assembly was and how precisely it differentiated the attachment site for the microtubules, which exist precisely at this end, from the chromatin and inner kinetochore binding sites at the bottom. So it really acts almost as a molecular ruler that provides a defined distance from the inner kinetochore to the outer kinetochore. And this is also visible in 2D class averages from the cryo-EM analysis of how rigidly this structure is assembled on the grids. And there are some other interesting features of this assembly which I don't have time to go into, such as autoinhibition mechanisms and so on that we observe in our structures. So effectively, from the beginning of my PhD to the time now, we could go from very good but descriptive understanding of how the human kinetochore assembles to something that is much more atomic in details and really understanding how all these components fit together. And I should also mention that my work really builds on a lot of foundations laid by other groups, and such a detailed mechanistic work will be impossible without everyone in the field. So far I've been talking about effectively the plus ends of the microtubules, but this is only effectively one half of the mitotic spindle. So then I wanted to share a story about the minus ends of the microtubules in mitotic organizing centers. Effectively, to generate microtubules, they need to be nucleated. And this is done at mitotic organizing centers or centrosomes in eukaryotic cells. And this is done by this ring-like complex at the minus ends of the microtubules, known as a gamma-tubulin ring complex, or gamma-toric for short. And a proposed model for how this happens is that gamma-toric assembles a template to which alpha-beta tubulins bind and be nucleated. However, there were two potential issues with this model. One is that the available structures, so information about gamma-toric, show that it's actually a very imperfect template. It exists as a very wide conformation structure. And the second is that actually purified or individual gamma-toric in solution are very poor at nucleating microtubules for reasons that are known. So the questions that we are trying to address there is, how does a gamma-toric nucleate microtubules in actual cells? To do this, we've purified yeast centrosomes, also known as SPBs, and collected many, many tomograms. And in these tomograms, we see that main SPB body nucleating or bound to the many, many microtubules. Then we selected all of the minus ends of the microtubules, as well as microtubules themselves, and performed a subtomogram averaging. This allowed us to determine a sub-nanometer resolution reconstruction of the gamma-toric complex, which is the ring-like structure, colored in blue and purple, sitting at the bottom, bound to the first few rows of alpha-beta tubulin. This had a few surprising, interesting features. One is that, first of all, in our reconstructions, when gamma-toric nucleates microtubules, the symmetry of the gamma-toric matches precisely and perfectly the symmetry of alpha-beta tubulin, suggesting that it is really a perfect template when it nucleates microtubules. The second striking feature of our reconstruction was this density shown in gold, which cannot be attributed to any of the known interacting subunits that was not known in the literature. And this density in gold really binds to every subsequent protofilament of the gamma-toric complex. We performed subboxing to improve resolution around this region, and we can really see that this is a very long coil that spans the gamma-toric. And if you take a side view of this structure, we see that this coil actually spans the base of the gamma-toric proteins, SPC98 and gamma-tubulin, and then effectively goes through the alpha tubulin and then binds the beta tubulin, effectively directly stabilizing attachment of the first alpha-beta tubulin to the gamma-torque ring. So with this data, we could propose a much more nuanced model of how microtubule nucleation happens in actual cells, where we have gamma-torque being in different conformation potentially, recruiting the coil-coil density because of its extensive interface with gamma-torque subunits. And coil-coil will have some affinity for the free alpha-beta tubulin in solution. It's effectively gamma-torque together with a coil-coil generating a partial initially attached alpha-beta tubulin ring, which creates a very high affinity binding sites between each alpha-beta tubulin subunits, which we speculate will be very, very quickly filled by free alpha-beta tubulin. So overall, we're effectively generating a ring of alpha-beta tubulin bound to the gamma-torque, which in literature and in previous studies been proposed to be the key rate-limiting step in microtubule nucleation. So in actual cells, we have these multiple factors that come together to nucleate microtubules very, very efficiently. With this, I just want to leave the final molecular model of how we think nucleation happens in the cells. And of course, this work will not be possible without a lot of support from everyone in the David Buffer's group. And a special thanks to David for being an incredible mentor and supervisor. And thank you so much for your attention.